All right, so you can.
Sorry.
All right. Hello, everyone. Thanks for coming, and it's a pleasure to give this talk and kind of interact with this club. So it's great to give a little bit of an idea of the kind of research that I'm interested in. So my name is Henry Shum. I'm an assistant professor at the Department of Applied Mathematics, and I'm going to be talking a little bit about some sort of fluid flows that relate to microorganisms and microrobots. So I think this is a really interesting topic, and it's something that's a little bit unfamiliar to most people. So hopefully it'll give you a little bit of inspiration and a bit of understanding about how things are a little bit different when you look at very small scale objects and things that are moving in fluids. All right, so just kind of navigating my PowerPoint. Hold on. All right, so I'm going to switch to my laser pointer. All right, so I'd like to start this talk by going a little bit back in time to something that's probably before your time and even before mine. So this was a really old movie from 1966. It's called The Fantastic Voyage. And the idea or the premise of the story is that I think there's a scientist or a doctor or someone who has some health condition, and the only way to save him is basically by miniaturizing a team of other scientists and injecting them into this person's body. And then there they can carry out some sort of procedure to, I don't know exactly what it was, but like remove a clot or something like that. So it's a really interesting idea. And it's kind of futuristic in a sense, because even though it was made in the past, we're still trying to get there, where we still have this inspiration, this goal or ambition to implement these ideas, not exactly in the literal sense of miniaturizing scientists or doctors so that we can be injected into other people, but by making robots and things that we can inject into people, or by making new kinds of nanomedicine and things that we can swallow, for example. So this is actually starting to become reality in the sense that lots of people have been working on this in the past couple of decades. And the main goals that people are interested in are things like targeted drug delivery. So we might want to, instead of administering a drug that affects your whole body, remember lots of drugs are very harsh, and it's not good to have these drugs in your body except when they're necessary to fight a particular disease. So you can target your drug delivery by making it release right. For example, if it's a tumor, you want it to release right inside the tumor and not just drift around the rest of your body. Other things that you might want to do are micromanipulation. For example, if you have, not necessarily in a medical setting, but if you want to assemble tiny things in like a micro scale factory, you can kind of use these robots to do this manipulation and put things in the right place. So there are lots of things like that. And here are just a couple of examples of real experimental things that people have done. This is an idea based on magnetism. So one of the most popular methods of controlling these microrobots so far is by using an external magnetic field. So if you have a magnetic particle, then as you should all know, a magnetic particle in a magnetic field will experience some forces and torques. And so you can use this external field. So for example, if you put your patient into an MRI, then that's one way of setting up a magnetic field. Or if you use some other kind of fancy coils, electromagnets to induce a magnetic field, then you can control these things that are inside your patient. Another way that people have kind of made micromotors is by chemical reactions. So instead of having mechanical things that are moving around or external fields that are forcing your object, you can use some sort of reactions, chemical reactions, to generate propulsion through various effects. So in this picture here, this is basically using bubbles that are produced by this reaction, and these bubbles come out of the end, and it's basically like a really tiny jet. So it generates a stream of bubbles that will propel it, and this kind of motion is very quick. And here it says stomach. So they've put this into, I think it was a rabbit stomach, and made it actually penetrate the tissue. So the title of this talk also mentions microorganisms. So what about those? Well, one of the first studied microorganisms in as far as mathematical modeling goes is sperm. And so sperm are very interesting, and I think you are probably most familiar with human sperm. You might have learned about that in school, in high school. But you might be surprised to know how varied sperm can be across different species in the animal kingdom and beyond. So here are a few pictures of different kinds of cells, sperm cells. These are drawn and these are images. But you can see how strange they can be, their shapes, and they're not like the usual kind of thing that you might picture when you hear about sperm. So all of these things, I mean, they have specialized features, and it's really an open question about why they have evolved in a way that they have all these very different structures and shapes, even things that are closely related to us, like rodent, like mice. They have very different kinds of cell shape, and there are also lots of different abnormalities that can be found. So one of the reasons people have been interested in studying locomotion sperm is the problem of fertility, or rather infertility. It might be surprising to know that, because if this hasn't crossed your mind, there's a very high prevalence of what we call subfertility or infertility, and that is basically the condition where couples who are trying to conceive have difficulty doing so. I think according to the Canadian government website, the current statistics for Canada is about 1 in 6 couples are what they would consider infertile, and the similar numbers would also echo around the world in other countries. Another thing that you might not be too aware of is when we talk about infertility problems, a lot of the times it's not the female, it's not a problem with the female. It can be also due to the male, so roughly half and half, if you wanted to make a guess. And so it's important to understand some of the causes of infertility. And one of the things that has been most focused on is the actual motility of the sperm. So are these cells able to get to the right location and do the fertilization? So people have been doing, in terms of clinical treatment and diagnosis, people are most often interested in sperm counts. That's the number of sperm or viable sperm per in the sample. But you have an issue not only with the number or the dense concentration of sperm, but also whether or not they are healthy and moving properly. So there's, in the media, it's been called a male infertility crisis, which sounds quite dramatic, but I think at the moment we're not trying to, there's no reason to be too alarmed at it, about it, but the idea or the problem basically is that it's been observed that in the last 30 years or so, the sperm count has been dropping quite a lot, so like 50%. Lower than your grandparents, for example, in terms of average sperm count. And this is a trend that's been going on for quite a while, and it's not quite sure, it's not quite known why it is, but this has been obviously also resulting in more cases of people having difficulties or seeking help with fertilization. So okay, so that was a little bit too much focus on sperm, I think. The other thing that's kind of interesting to me personally in terms of research is bacteria, because I've been focusing more on simulations of bacterial swimming. So here is a brief movie of a video of some bacterial cells swimming around. So you might also have seen movies like that, or you might have an understanding of how bacteria swim already. They basically have a long flagellum that they rotate around, and that kind of acts as a propeller. And just before I move on, because later we'll come back to sperm. Sperm often, most of them will also swim using flagella, which are these long structures. They're very different from bacterial flagella, but for our purposes, you can think of them as kind of filaments as well. And they have some motors in them, so molecular motors that actually cause them to bend. So they can propagate waves, and that's the kind of familiar sinusoidal traveling wave pattern that you might have seen before, that helps them to swim, or that makes them swim. All right, so I forgot to mention these other examples like microalgae that swim. They're very important because, because they're also everywhere, phytoplankton and lots of stuff in the ocean. And lots of diseases are caused by parasites that are microorganisms that swim around in fluid. They can get transmitted, for example, through mosquitoes or in the unsanitary water and things like that. So there are lots of unpleasant applications of microfluidics and microorganisms. Okay, so what do they have in common? Well, basically, the way that we would try to analyze their motion, or the way that we would try to mathematically describe the governing equations, is by using an equation for fluid flow. And for physicists, the usual equation of choice is the Navier-Stokes equation. And this describes most common kinds of fluid flow that we might be interested in, from water to air as well, and things like that. So this is basically an equation about momentum conservation. The right hand, let's see. So just to kind of clarify, in case you haven't seen, if you're not too familiar with these kind of vector equations, U is a vector, so it has three components. And if we have three spatial dimensions, we have Ux, Uy, and Uz. And it's a vector field, so the velocity, U is the velocity vector field. And it depends on position, so X, Y, and Z coordinates, and also on time, because in general, things can change in time. The notation, the nabla, or the gradient, I hope you're familiar with vector calculus. So the gradient term is going to give you a bunch of partial derivatives of our flow field. And since U itself is a vector already, the gradient becomes a tensor. So you have basically a matrix of partial derivatives if you use this Cartesian reference frame. And the grad squared is the Laplacian of the velocity field. Okay, so what do all these terms mean? Well, let's start from the left-hand side. So rho is the density of fluid, and we're going to assume that rho is constant. So this is a fluid that doesn't change in density. We don't care about things like expansion with temperature and compressibility. So going along with that, we have an additional equation. So you'll notice here we have actually two equations. One is this thing here equals the right-hand side, and then we have a second equation, which we call the incompressibility condition or constraint. And this is the divergence of the flow field equals 0. So it's a divergence-free flow field. And what that basically means is that you don't have any sort of accumulation, or you don't have a source or a sink for mass. So basically, the fluid can flow, but it cannot accumulate at one particular point. So the density doesn't change anywhere. Okay, so the fluid has to keep moving. And this condition basically means that your velocity flow field, even though it's a vector equation, it's a vector function of three spatial coordinates, they're kind of linked together. They have to satisfy this constraint, or this incompressibility equation. So the components Ux, Uy, and Uz they have this relationship. Okay, so going back to the equation, I said that this is a conservation of momentum equation. So what do we mean by momentum? Well, this is basically Newton's second law. So we're familiar with force equals mass times acceleration. And so for a fluid, we don't really care about the total mass because, well, that depends on how much volume you have. But rho is the density, so it's a mass density, mass per unit volume. And then this term in here, which we call the kind of convective derivative of U, it's basically combined, this gives you the inertia per unit volume of the fluid at a particular point. So this is the partial derivative of U with T. That's like you can think of that as acceleration. But then this is a little bit more complicated because you have this other term, which is basically the rate of change of So if you follow a piece of fluid, a particular parcel of fluid that has a fixed collection of molecules, then this collection of molecules is moving with the flow. So even if your d partial u partial t is 0, that means your flow field is constant. That parcel of fluid can change in momentum, because as it moves around in your vector field, it will be in a different position, which means you can have a different velocity. So we want to actually track the rate of change of the momentum of a particular piece of fluid, and so we have to include that term here, which comes from this u dot grad u. So this is a nonlinear term because you have u appearing kind of twice as this product. So that's what we mean by nonlinear, and I'll come to that point in a minute. But basically, the left-hand side is inertia. It's the mass times velocity, or mass times acceleration, actually. So, so this is more like a rate of change of inertia. The right-hand side is the forcing term. So what forces do we have here? Well, we have one which is called the pressure gradient. And I won't really talk too much about the pressure gradient, but this basically, the pressure is something that that comes, it's kind of induced in order to satisfy incompressibility. So if you don't have this term, then it's not guaranteed that your vector field will satisfy this. So we need this pressure term. Basically, it acts kind of like a Lagrange multiplier, if you've heard of that. This is like a thing that constrains, that enforces your constraint that we're imposing. The main other force that we're concerned with is the viscous force. So here mu is the viscosity. It's called the dynamic viscosity, and this is usually a property of the fluid, and we can assume that it's constant. The Laplacian of the velocity field basically is how the changes, the spatial variation of the velocity field gives rise to viscous forces. So you can see that this viscous force kind of acts to reduce gradients or to reduce variations in your velocity. So it tries to smoothen out your velocity field. Kind of like a diffusion term, if you're familiar with diffusion processes. Okay, so as a mathematician, it's very common to try to non-dimensionalize or to want to non-dimensionalize equations. And that basically means that we want to rescale. We want to redefine some new variables that are dimensionless. And that means that we are able to check what the contributions of different terms would be under different circumstances. So if we have a particular problem, so if we have a spherical particle, the sphere has a radius r, and we're moving it. So for example, it's moving under gravity, or something is pushing it. So it's moving to the right with some speed u. Then we can rescale our variables. Our velocity scale will naturally choose this u. So that means we divide our dimensional velocity vector u by this scale, capital U. And this gives us a new variable that we call u tilde. And this is dimensionless. Okay, so it has the same role. It's still velocity, but it's in some sort of rescaled units of, so there's no dimension. X is the spatial coordinate, and so naturally we choose the length scale r that comes from our problem. So we divide all of our spatial variables by r to give us our dimensionless x tilde, and then time. Well, we haven't got a time scale here, but we can create one because u is basically a length per unit time, and r is a length. So if we divide r by u, then we would get something that has dimensions of time. So that would lead to our natural choice of rescaling by u over r. And so this is our dimensionless time. So now, in case you're not very familiar with the process of non-dimensionalization, basically we go back to our original equation and we do a substitution of wherever we see a u vector, we substitute in capital U times u tilde. And then we can pull out all of these scales so that they factor out. And then the idea here is that, let's just take the inertial term here. And that when you put everything in, put these new variables in, we have something in parentheses which looks basically like the original. So it has the same significance, the same interpretation, but we've pulled out some scales, rho u squared over r. And we can do the same for things on the right-hand side. So we'll just focus on the viscous forces. If we do the same thing here, then we get something that's now the Laplacian in the new coordinates of u tilde. And then we have the scale here. And what we're interested in, or what we can define, is something called a Reynolds number. And the Reynolds number is the ratio of the scale of the inertial force divided by the scale of the viscous force. So obviously these things in the parentheses, we don't really know. They depend on the problem and they depend on space and time. But we've chosen some scales that we kind of assume are representative of our problem. And so we divide this. This is the inertial scale divided by the viscous scale. And that gives us this ratio here. And so this is what we would commonly define as the Reynolds number, rho UR over mu. So again, rho is density, U is a characteristic speed or velocity, R is a length scale, and mu is the viscosity of the fluid. Okay, and this is important because it kind of tells us what is important in our equation. Remember here we have three main terms in our Navier-Stokes equation, but if the Reynolds number is small, that means basically the inertial force is not very significant, and that means we can kind of neglect the inertial term. And that's what we're going to do later on. I'll come to that. The other situation might be where the Reynolds number is very large, and that means inertial effect is really significant compared to the viscous effect. And so in that case, you might want to completely neglect the viscous forces, and that simplifies your equation by then just reducing to this, this balance of terms there. Okay, so let's compute a few Reynolds numbers. So for things in water, for a bacterium in water, the R, the length scale, it's about a micron. So bacteria are usually about a micron wide and a couple of microns long. So let's take R to be 1 micron or 10 to the minus 6 meters. They swim quite quickly for their size. It's about 10 microns to 100 microns per second. So this is our U. The viscosity of water is this number here, and the density of water is that number there. So if you put that into our formula for the Reynolds number, you would get a number that's about 10 to the minus 5. And of course, you'll notice that the Reynolds number is dimensionless, which is the whole point of a Reynolds number. That means that when we talk about small Reynolds number or small numbers, remember for a pure number, you can say that something is small or large, and what you mean is small compared with 1 and large compared with 1, small in magnitude compared with 1, or much larger than 1. When we talk about things that have physical dimensions like a length, it's not clear what we mean by small. So if you say that R is small, well, what do we mean by small? Is a micron small? It's small to us because we can't really see it. But if you were an atom, you would say that 1 micron is really large. So that's why it's very important and very useful to non-dimensionalize your equations that you get from physical models and from other systems. It gives you a way to interpret what we mean by small and large, because now it's very clear that small Reynolds number means that the inertial forces are small compared with the viscous forces. So we're comparing two things and saying that one is smaller than the other. Okay, so 10 to the minus 5 is the Reynolds number for bacteria. As an example, suppose we went out swimming. So it's been quite warm these few days. So if you went to the beach or went swimming in a pool, what would your Reynolds number be? So I'm not a very good swimmer, so let's say as a rough estimate, the speed might be about 1 meter per second. R, okay, so R can be any sort of length scale, and it's just an order of magnitude estimate. So it could be your height, which is say 2 meters, but you can round it down to 1 meter just to get an order of magnitude. Mu would be the same as the water that we have for the bacteria, and the density of water is the same as before. So just because we have a large length, large compared with the bacterium, and we are also much faster than the bacterium in terms of the absolute speed of swimming, we have a really large Reynolds number. So this gives us something like 10 to the power of 6. And so we are about 11 orders of magnitude higher in terms of Reynolds number than the bacterium. So that's going to be important in terms of how we would describe a human swimming compared with how we would model bacterium swimming. So just to give you a little bit of a comparison or something to think about, let's do one more example where we try a human swimming in a very viscous fluid. So pitch is also called tar sometimes, or bitumen. So this is a really thick, really viscous oil product. The viscosity here is 10 to the power of 8. And so that gives us the kind of 11 orders of magnitude difference that we need to bring our Reynolds number back to the same as a bacterium. So if we were somehow able to swim in pitch at the same speed, which is really unlikely, but if you were able to do so, then that would give you the Reynolds number that's comparable to bacterium. And that means that you could use the same equations, and the same flow field would kind of apply if you do the rescaling. So just as a little side note, out of interest, pitch is something that's so viscous that this experiment, which was started in the 1927, I think. So since 1927 or since 1930, when they did some adjustments, it's basically, this is a funnel containing some pitch that they put in, and there's a hole at the bottom so the pitch can flow out, it can drop. And since 1930, only 9 drops have actually fallen from this funnel. You can see the scale. This is a 9-volt battery, just to give you an idea of how large it is. But this is so viscous that it takes forever to kind of flow. And I think it's supposed to, they estimate it can last for another 100 years or so. Okay, so anyway, that's just kind of to illustrate how hard it is for a bacterium to move. So that's kind of scale, that's kind of problem it has to deal with when it tries to swim. All right, so as I said, the Reynolds number is small, and that means we can basically neglect inertia. That's irrelevant. And that means that we reduce the left-hand side to 0, and this is the equation for Stokes flow, or time-independent Stokes flow. So that's quite interesting. This equation is much simpler, and it has some interesting properties. So first of all, it's linear, which means that u only appears in a linear fashion. There's no u squared anymore that we get from that. And that means, as a linear equation, we can use the principle of superposition. So if we have 2 different solutions that satisfy particular boundary conditions, then we can add them together and we get a solution that satisfies the associated set of boundary conditions. So that's very useful to exploit. And in particular, we have some exact solutions that we wouldn't be able to do for many cases for Navier-Stokes flows. And this is, for example, a translating sphere. So the problem that I set up before, where we have a sphere radius r moving at particular speed, this would have this formula for the velocity field. And so u is a function of position. If we take the sphere to be at the origin, then this r is the position vector of the field point, the point where you're looking at the flow field. And this would be the equation that gives us the flow field, the flow at any particular point around that sphere. So here, this capital U is the velocity vector of that sphere. And you can see that it only depends on the distance. Small r is the distance away from the sphere. And it's kind of has this interesting dependence. So small r is the distance here. And the first term has a 1 over small r dependence. So there's a factor of 1 over small r there. U is a constant. And this term here, even though it has an r squared on the bottom, you can see actually that there's an r squared on top as this vector form. So this has a sort of order of magnitude, which is the same as u. So this whole term here has an order 1 over r, or capital R over r, if you want to make a non-dimensional comparison. And the next term here has a higher order. So you can see here that, well, this has the same order. Again, there's r squared on top and r squared on the bottom. So this is order 1. And the factor that we pulled out is r over small r to the power of 3. So actually, I'll have to check if that's correct. That might be 1 over r squared, actually. But anyway, this is a higher order than the previous one. And so when you're very far away from the sphere, The most important term is this. If you think of sort of expansion, this is a small number because 1 over r is large when you're far away. And this is even smaller because it's 1 over r cubed. So you can neglect this term in the far field. Okay, so this term here, because it's so important, I'm going to give it a name. This is called the Stokes slit, and this represents the flow due to a force. So if you put a force in a particular point in the fluid, and you look very far away from that point where you're applying the force, then this is basically the flow field that you would get. And then, as I said before, you can use superposition, which will come again. We'll come to again. If you put lots of forces at different positions, you just add up their effects, and that gives you the flow field due to that collection of forces. Okay, so here's a really classic video demonstration from, this is taken from YouTube, but this is an experiment done by G.I. Taylor, and this is demonstrating one of the properties of low Reynolds number flow, which is called reversibility. So let's see if this works. Oh, sorry. So here he says some ink that says R much less than 1. R is the Reynolds number. And then he turns this handle. The handle is connected to the center, so it's rotating the fluid. And this thing here is the fluid, but it's very viscous. So I hope you heard the audio. Did you hear the audio? So basically, the demonstration is that if you move your system, move your fluid forward by moving the boundary. So in this case, it was the inner boundary. Let me see how I can pause this. Okay, it looks like when I pause it, it has this banner. Okay, that's unfortunate. Okay, so basically, when you turn the handle, it moves the inside, and the outside is fixed. So it's a cylinder of fluid, and you move it around. It's basically like stirring. If you have a coffee cup and you stir it, but you're only stirring by rotating the inner cylinder. Now, initially there was some dye that was had some writing on it, and if you and so when you stir it, that dye obviously is going to move. It's going to spread out, and it looks like it's basically mixed. But the interesting part in this demonstration, you can see here, is kind of destroyed. When you reverse it, because the equation basically doesn't care about the speed. And then you get back the original pattern of the ink that was given into the fluid. So what this demonstrates is something that was called the Purcell Scallop theorem by Purcell. He wrote this very interesting paper, well, based on a lecture that he gave. And basically the theorem, and this is the only theorem of my talk, the theorem says that a scallop cannot swim at low Reynolds number. So what do we mean by a scallop? Well, maybe you know what a scallop is. It's a kind of seafood, usually. It's a clam, or it's related to a clam. So it has two pieces of the shell that are hinged together. So this this thing, so I'll use my hands here if you can see my video. The shell can open and close, but that's all it can do because it only has one hinge, 1 degree of freedom. And from the video, what we saw was that if this is moving in low Reynolds number, when you close it, you might move somehow. Maybe you think that you're pushing the fluid forward, so you move backwards. And so that might be a way of swimming. But if you then have to open yourself, then that's basically turning the handle backwards in time. And that will mean that you're actually pushing yourself back into back to the original position. So if you keep doing this reciprocal motion, you're only going to wiggle around a little bit, but you don't actually get anywhere. On average, the net displacement will be 0. So that means you're not able to swim. And that's very important because it means that there's a restriction on the kinds of motion that you can use in order to actually to get anywhere to swim. So if you imagine, if you learn swimming, there's one of the most common style is front crawl, or sometimes called freestyle. And so if you think about the leg motion that you do, you're basically alternating your legs, and that's basically a scallop, because you can think of your legs as doing this kind of motion, even though, well, they're actually crossing over each other. But you're doing a reciprocal motion. And if you thought that that was tiring in a swimming pool for your Reynolds number, for a scallop, that would be even more futile, because you would just keep going backwards and forwards and not get anywhere at all. So, so how can we get over this? Well, this is only for precisely reversible or reciprocal motions. So it turns out that, in fact, if you have a slight bend, if your legs are not perfectly straight, or if your legs are slightly flexible, then that would be enough to evade the Scallop theorem, because then your motion, although very slightly perturbed, is not exactly reciprocal. And so it would be possible to have some net displacement if you have some extra degrees of freedom, like bending. Another way that was proposed by Purcell is to have not just one hinge where you have a scallop with 2 pieces, but if you extend that to 3 pieces with 2 hinges, so now you have 2 degrees of freedom, because you have an angle that describes the first hinge and an angle that describes the second hinge. And that would be enough to allow some net motion if you do some sort of periodic deformations of theta 2 and theta 1. So in this lecture, Purcell proposed the situation where you kind of open one, you move one of the hinges down, and then you move the other one down, and move the other one up, and then the other one up. So this is a sort of cyclic pattern of the opening and closing, or moving up and down these arms. Okay, so well, this is just a mathematical model for some sort of motion. It's not like not the way real scallops would swim in the Reynolds number, but you can think of this as a very coarse model for sperm. For example, if you remember, the sperm had this kind of sinusoidal wave pattern I mentioned before. So it's like a flagellum that propagates some bending waves. And if you extend this, so if you add on even more links and more hinges, then you can think of this as a collection of things that are hinged, and they can bend. And that kind of approximates a filament that can bend along its length. So, so the same sort of principles would apply as long as you have more than 2 degrees of freedom, such as a continuous deformation. All right, so another way that you can think of some very simple idealized models of swimming are things where you have 3 spheres. Instead of 3 hinges, consider 3 spheres that have some sort of very thin arms between them. So these arms can, you can think of them like pistons. They expand and contract in some periodic fashion. And again, this is this allows you to circumvent, overcome the Scallop theorem, because you have 2 degrees of freedom. You have 2 arms that are controlled independently, and so you can calculate basically. Remember, I gave you the solution for the flow field around a sphere that's translating. So at any given time, you can think of the forces that are acting on all of these spheres, and the flow fields that each moving sphere is generating, and how that will affect the other spheres. So it's not a very complicated problem. You can kind of think about this yourself, see how far you get along. But at least conceptually, it is quite simple. But working on the details is a challenge. So you can look at this reference if you're interested in how you would calculate or approximate the velocity or the net displacement that you get. So this is after 1 cycle of motion, you have some net displacement in the positive x direction. And that'll depend on the amplitudes of your oscillations and things like that. Okay, so let's look more at a biological example. Well, in case you're not too familiar, bacteria, flagellated bacteria usually swim by rotating their flagella. So unlike the sperm, which I mentioned before, the bacterial flagellum is actually just a passive filament. It's kind of like a wire or a rope. It's slightly rigid, so it can hold its shape basically. And it's usually kind of corkscrew shaped. And so you can think of this, well, like a corkscrew. If you turn the corkscrew, it's going to move into the cork Similarly, it's going to move through the fluid. So the idea here is that we have a rotary motor. And this is quite amazing because remember the cell itself is only about 1 or 2 microns long. And you can see here the scale of this is 20 nanometers, 27 nanometers. So this is a very tiny molecular scale machine, but it can rotate just like a rotary motor that you might have in, say, a toy car or something like that. And so this motor turns a structure, the rotor, and that structure called the hook is connected to the filament. And so it's going to turn the whole thing. And that's how you get your propulsion. Okay, so this is just a couple of videos, examples of how bacteria swim. And you can see here the kind of helical shape of the flagella, and again here. But you can see that when they stop, things can go kind of strange, so they can change their shape a little bit in terms of the flagellum. Okay, I won't focus on that too much, just to give you an example. And there are lots of other weird and wonderful examples in nature of different kinds of organisms that can swim and create fluid flows at microscale. So here, this is a kind of flagellated alga or algae. So this is photosynthetic, but it has 2 flagella. Let's see if we can see the video here. You can see these 2 flagella are kind of waving around, and this is what people call a breaststroke. So it's very similar to what a human would do as a breaststroke. So I said before, if you do the kick for the front crawl, that's not going to work. But if you do breaststroke, that would work because it's not reciprocal motion. Even though you're sort of opening and closing your arms, there's a lot of curvature, a lot of bending. So the forward stroke, if you like, is different from the reverse, and so it's not reciprocal. It's not reversing its motion. Then this movie down here, I'm going to give you a full screen because it's really fascinating. So this is the flow field around a starfish larva. So here they've kind of fixed it to a substrate, so it's not able to swim around freely, but it's generating these fluid flows that you can visualize by imaging particles that are moving in the fluid. So that's really interesting, isn't it? This is much larger than bacteria and other things, by the way. All right, so how could we go about analyzing or predicting the motion? Well, I'm kind of running over the time that I was expecting, so I'm not going to go into much detail, but the idea, one of the key ideas that people have used for its simplicity is called resistive force theory. And this is basically founded on the idea that because of the linearity of the Stokes equations, we can sort of sum up the contributions from little elements of our filament. So this only applies, resistive force theory mainly applies for slender objects like a very long, thin filament, the flagellum. And the idea is to conceptually cut it up into small pieces like you would in basically the idea of calculus. You consider an infinitesimal element of your length, of your flagellum, and look at the forces that are acting on it. And if you look at a small piece, then that looks kind of like a straight cylinder. And for a rod or a cylinder that's moving through fluid, you can basically use the linearity of Stokes flow to come to the idea that the forces and the velocities are proportional to each other. So basically, if you exert a force in the, say, the tangential direction, then it's going to give you a velocity in the tangential direction by symmetry. And similarly, if you exert a force in the normal direction that's transverse to the axis of the cylinder, you get a velocity, a reaction force. So maybe I said that wrong. If you move it perpendicular, then you get a force that's perpendicular to the velocity, to the motion. And these are linearly related. So we can express this basically as saying the tangential force is proportional to the tangential velocity, and the normal force is proportional to the normal velocity. But an important fact here is that the coefficients of proportionality are different in these 2 directions. So CT and CN can have different values. It turns out that CN is usually larger than CT, and the ratio is kind of 2. So CN is roughly twice as large as CT. And this means that it's more difficult to push a rod parallel tangential, like in the normal direction, than it is to push it along its length. And that, I think, kind of makes intuitive sense. It's the same as if you had a flipper and you're kind of moving it with a broadside. That would give you more resistance than if you tried to slice through the water tangential to your object, to your flipper. So this is actually fundamental because it means that when you consider the motion of any piece of your slender object relative to some background fluid, it will give you a force that is not exactly in the same direction as the velocity. So here we're assuming v is in the vertical direction. Your resultant force, your drag force, is not exactly vertical. It has some horizontal component. So if this is your sperm, and it's doing some sort of motion, you can see here that this motion, so it's sinusoidal, so you have parts that are moving up and down, but basically you have some horizontal component of the force, and this gives you, if you sum up over the entire flagellum, that would give you the net force, and this is like a thrust because it's moving in the horizontal, it's a force in the horizontal direction. And in order to balance your forces, you're basically going to have to move your object to the left. So by doing some sort of motion like this, you can generate a horizontal net motion of your object, and that's what we call swimming, right? Okay, so so that idea is simple. You can kind of write down a model, at least conceptually, about how that's going to work, but it's very limited in accuracy because you're sort of treating each piece of your filament as independent and in isolation. Well, that's the same thing. But what I mean is they don't affect each other. So you're not actually solving the fluid, the equations of Stokes Flow. We're not satisfying any particular proper boundary conditions. We're just saying that each piece sort of has a drag that depends on its motion relative to some background zero velocity flow field. Which is not true because when you have some parts of the flagellum that are moving, then that would disturb the flow field, and so you have to really take into account these interactions. And moreover, if you have something like bacterium, there's a really large cell body attached to the flagellum, and that cell body is going to perturb the fluid flow as well. So, so resistive force theory kind of neglects all of that. It's still quite useful. It gives you a rough estimate, and it can give you some really interesting results, but it's not an accurate way to deal with interactions. And so a better way would be based on this idea. Remember before I gave you the solution for the flow due to a force, I called it the Stokes slit. So if you imagine a surface of your bacterium, so I haven't got a picture of one, but if you have any sort of organism and you cover it with forces, then you're going to have a flow field that's generated by this, distribution of force. And so that's basically a generalization or a way of constructing flow fields that satisfy Stokes equation. And that's much more flexible, much more general. And this way you can include things like walls. So for example, if you're a sperm, you're not going to be swimming in infinite fluid. You're going to be swimming near some channels and some walls. So it's important to take into account these other walls or other swimmers or other particles in your system. So here's a quick overview of how you might come up with a simple model for bacteria. So now we're switching to bacteria because they're simpler kinematically. You just have this cell body that's rigid and a flagellum that you can assume is also rigid. And you just impose some sort of frequency that the motor is turning this flagellum relative to the body. So our objective is to calculate the overall swimming velocity. And that means U is the translational velocity of your bacterium, and omega is the rotational velocity of your bacterium. So, okay, so this is what we have to figure out. And well, so I drew this as a kind of triangulated mesh. So hopefully you've seen something like this before, a mesh of a surface. Basically at each node, each intersection or vertex of the triangles, for example, you can assume that we're putting a force at these positions, at these nodes. So this is a way of discretizing our system. Instead of having a continuous boundary condition, we just assume that we have these different particles, different points along our surface. So we have, again, our Stokes Flow equation, and then we want to satisfy the no-slip boundary condition. And the no-slip boundary condition means that the fluid flow is moving, or the fluid is moving with the same velocity as the bacterium at the surface. And this is a very common assumption in fluid mechanics, because basically it's saying that the fluid sticks to your surface, which is in good experimental agreement. So what happens here is that we want to satisfy these boundary conditions and our governing equations. And we do that by expressing basically the flow field as an integral, or you can think of this as a sum over discrete points of these forces. So these forces are the forces that are being exerted on the fluid at each of these points. And G is what I called the Stokes up before. So I didn't give you exactly the formula. I gave you the formula in terms of the velocity of a sphere, but you can think of it essentially in the same form. It has a 1 over r dependence. And so basically this is a sum of contributions of forces from different points along your surface. And you add them up by linearity, and that will give us the flow field at a particular point x naught, which is an observation point somewhere outside or somewhere in your domain. Okay, so the problem here is that the forces are kind of undefined. We haven't said anything about what the forces are. That's not part of the boundary conditions. And so what we have to solve is, so we have to solve for that as well. So remember, we want to find the 6 velocity vectors, 6 velocity components. If we have n mesh nodes, suppose we have 3 n traction is a force. We have 3 because it's 3 dimensions, so 3 components for each node. So we have a bunch more unknowns now. And we only have the boundary integral equation, which gives us an equation for each point on our mesh. So that means we have 3 n equations. We also have this condition where we want to satisfy something called no net force and no net torque. And this is basically the statement that, remember, we're in the Stokes regime, which is inertialess. And so if we have no inertia, that means that the net force is 0. F equals ma equals 0. That means the net force is 0. So the total amount of force that we're exerting on the fluid must be 0. And the total torque, which is the cross product, x cross f, that should also be 0. So this is a vector equation, that's a vector equation. So we have 6 more equations here. And luckily, we have the right number of equations as unknowns because we have 3n plus 6 unknowns. We're only interested in u and omega, but we have these other unknowns as well. And then we have a bunch of equations. And it turns out that we can solve this system of linear equations just as you would. It's a matrix equation like ax equals b. You just invert that or you solve it, and then that will give us the u and omega. So that tells us the velocity of our swimmer. Okay, so I've gone a little bit over time, but here are a few quick simulation results. So in the experiments, I didn't really talk about the tracks, but what we see commonly for bacteria is that they swim in circles. And this actually only happens because when we're looking in a microscope, we're looking usually close to the slide, close to the glass slide, and the interaction of the bacteria with the slide causes this curvature. So if you don't have any slide, then the bacteria can swim straight. There's no problem with that. But because of the way that the bacteria swim, it has a head that's rotating and a tail that's rotating. So what happens here, let me just skip that. What happens here is that you can think of, this is now different views of the bacterium that's swimming in this kind of circular path. If you look ahead of the bacterium from the front, the cell body is rotating a particular way because that's how it moves. And the effect of the wall kind of resists the motion of this rotating circle. So it's kind of like rolling a ball along the ground. If you roll the ball this way, it's going to move this way. So the head, the cell body wants to move in the left direction. But now if you look from behind the tail, the flagellum, the helix, is rotating in the opposite direction because the motor is turning it relative to the cell body. So if this thing is turning, then again, you have this effect of the wall below. It wants to make the helix roll to the right. So here, the head is rolling to the left, the tail is rolling to the right, and so overall it's going to turn. So it turns as it moves forward, and that gives rise to this circular path. So this is all due to hydrodynamics, and you can sort of calculate that, or you can use some numerical things to work out the radius of curvature and so on, and that's kind of what we see in the simulations. Okay, so in the interest of time, let me just skip a little bit of that, but I want to go back to the idea of microrobots because I think that was kind of one of the interesting motivations for this talk, for this subject. And so just to give you an idea of what how far we've come in terms of microrobots, there are some attempts of making scaled down versions of a bacteria, like artificial bacteria. So this is like a little body piece with a motor, something that's turning. And then you have a rigid copper wire that's turning here. And so that gives you the same effect as my model bacteria. The issue with that is that this sort of thing is quite large. It's not the scale of a real bacterium. It's about 50 millimeters long, compared with something more like 5 microns long. But there are obviously challenges with miniaturizing this. You can't, so far we don't know how to make batteries small enough and motors that are small enough to fit onto a micron size object. So another alternative, if you don't want to worry about making a motor, you can actually use an external magnetic field. So if we just have something like the helix without the motor, then obviously that's not going to swim by itself because you need something to turn it. But if you attach some magnet to that motor, to that helix, and then use a magnetic field, then you can turn this magnet by rotating your magnetic field, and that's going to in turn make the helix turn. And it's this helix, the coupling between the rotation and the translation of the helix that's going to make this propel, just in the same way as a bacterium would, except in this case, the cell body is fixed to the tail. And so this was demonstrated quite a while ago, 2009. They used a magnetic field and they could steer it so that it writes out some letters or some whatever pattern you want it to move along, whatever path you want it to move along. And some other examples, there are many, but here's one other example where they have something more like a screw. So this is essentially it looks, to me, it looks like a screw that you can find at a hardware store. It's magnetic, so again, you use the magnetic field and you can make it penetrate what they say is a bovine tissue sample, which to me sounds like they just got a piece of steak and played around with it. But that's quite interesting because then that gives us, that puts us in the realm of using these things for medical procedures or things that would, where they interact with real tissue and they can travel through and go to the right place. Okay, finally, there's some other examples like here, this is called a Spermbot. Let's see if that works. This is basically the same idea. So we have a magnetic field driving a magnetic helix. And here there are some sperm, and you can see that the sperm are not very happy, they're not swimming. And the idea here is that we can use these artificial devices, artificial micromotors, to capture, like we grab onto a sperm and push it, and you can push it actually into the egg. So that's really cool, really interesting. Of course, you would need the sperm to be functional, so you want it to actually be able to fertilize. But if the only issue is in motility, then that's conceivably an interesting solution. Then there are other things like using bacteria, attaching it to a payload. So now we're kind of harnessing real like existing motors, existing organisms that can swim, and using them to propel our payloads around so that we can deliver drugs to particular locations, assuming you can control where these bacteria swim. All right, so I just want to end with a video that I won't really comment about, but this is a simulation I did about some bacteria interacting with other particles. And so basically the conclusion that I would like to leave you with is that microorganisms are really interesting and fascinating, and they can inspire a lot of creative ideas and microrobotics and stuff like that. And being in the Reynolds number, although it's kind of unfamiliar to us, it really simplifies some of the equations and allows us to solve them in ways that we can't for larger flows. And we can use these models to kind of help us understand and predict the behavior of microrobots. And there are lots of interesting ideas that you can come up with and examples from nature to inspire new designs. All right, thanks a lot for your time, and I'd be happy to take questions now.
Thank you so much, Professor.
You're welcome. It's my pleasure.
I had a question about the the front crawl example, and later when you were talking about the effect of the walls, and it just made me think of like the effect of the walls in a swimming pool. I was curious about the effects that the walls of the swimming pool have on the simple example of the front crawl.
Yeah, that's a very good question, and that's very relevant. So what you can see actually in this video is that if you were in a low Reynolds number environment, so if you had a bacterium and you swim around, you're actually dragging a lot of fluid around with you, even though you can't really see the fluid motion here. You can see that this particle that's in the fluid is moving because of this bacterium that's passing by. And the way that particles in the fluid move in the Reynolds number is a much longer range interaction than you would be used to for yourself swimming in a pool. So for example, if you were in a pool moving around and swimming, you don't expect to be moving your friend very much if your friend is swimming in the lane next to you. But if you were a bacterium swimming, you would be actually heavily influencing the motion of other swimmers, other particles in the fluid. And the converse is that the walls have an effect. So in electrostatics, there's a kind of idea of image systems. So if you have a charge and you want to satisfy some no charge condition on a boundary, you put an opposite charge on the other side of your boundary, and that sort of cancels out the effect of the original charge. And that's one easy way of coming up with a solution for a different geometry. In Stokes Flow, you have something similar. You sort of have a negative swimmer. You have something that cancels out the flow due to the original swimmer on the other side of a wall. And so you can think of this as being swimming near a ghost, near some sort of negative swimmer, parallel and opposite to you. And there's a sort of attraction depending on the kind of fluid flow that the original one is producing. There will be some interaction. So it's like you're being attracted to the mirror image of yourself. And so these are kind of interesting observation, some interesting results of the long range hydrodynamic interactions for humans. In the high Reynolds number regime, we sort of neglect, or we can neglect the viscous effects except in a very thin boundary layer. So very close to the surface of the object, you do have some viscosity. So things are not completely inviscid, but in the rest of the fluid, we would use the equations for inviscid flow, which means that we drop out the viscous term. And that means, and the effect of that is that we have much less of a influence on other things that are in the fluid, and especially things that are kind of in front of us. So if you think about a boat that's swimming, a boat that's moving through water, or a submarine, you can probably imagine that the fluid just behind it is quite different from the fluid just in front. There's a strong asymmetry, and in particular in front of it, there isn't going to be too much disturbance flow. So it's like the effects of the submarine it wouldn't be felt in front of it very much. Whereas directly behind it, you would kind of feel some turbulence and some perturbations. Whereas for low Reynolds number flow, it's much more symmetrical. It's not exactly symmetrical because the fluid flow might not be symmetrical, but you can push things in front of you just as easily as you can kind of pull things behind you. And so what's interesting here is you can see that we'll have to wait for it to come back around, but you can see that initially this particle is being pushed ahead of the swimmer because the head of the bacterium is basically pushing the fluid, and that means you're pushing the particles. But when it reaches this point, it's kind of attracting the particle into the swimmer, and now it's pushing it backwards. So the tail is kind of pushing the fluid backwards, and that means that you're moving a lot of fluid backwards. But then you also have some areas where you're pushing the fluid forward, and pulling things in and away from you. So things are quite interesting and more complicated, I think, in this kind of interaction.
Thank you.
If there are no more questions, then I guess that's it. Evan, do you have names? Do you want to announce that now or later?
We can do that now. We have 4 that 4 gift cards are given away and 4 people entered. So if you message me, you know who you are. You're getting food. And Professor, as some thanks for your coming here today to give your talk, we'd like to give you a gift card too.
Oh, thank you very much. That's very kind.
Yeah. So I guess like one of us already has your email, so that's taken care of already.
Yeah.
Okay.
We'll figure that out.
Yeah. Thanks so much for giving a talk today, Professor.
Great. Thanks for listening, all of you.
Yeah,
Thanks for the talk. It was super cool.
Thank you.
Okay, so I'm gonna stop recording then.